This video is about development of cornea. To understand how the cornea develops, one should have understanding of development of eye. To understand development of eye, one should have basic understanding of general embryology, the beginning part of it. So to understand what is neural plate, what are three germ layers, how the neural tube forms, all these things can be well understood by having the basic understanding of general embryology. For that, I have recommended one 19 minutes video on the last slide. The eye is derived from two main germ layers, ectoderm and mesoderm. So ectoderm is divided into two parts, surface ectoderm and neuroectoderm here, and the mesenchyme is derived from neural crest cells or mesoderm. Ectoderm is divided into two parts as I have mentioned, surface ectoderm and neuroectoderm. The surface ectoderm, it gives rise to lens, epithelium of cornea, conjunctiva, lacrimal gland, lacrimal apparatus, eyelids and its derivatives. Neuroectoderm, it gives rise to retina, ciliary body epithelium, iris epithelium, sphincter and dilator pupillae muscles, optic nerve, its neuroglia and nervous elements only, but the secondary vitreous and tertiary vitreous which corresponds to vitreous base and ciliary zonules. The mesenchyme that is derived from neural crest cells, it gives rise to endothelium and Descemet's membrane, the most part of the sclera and choroidal melanocytes. The mesenchyme that is derived from mesoderm, it gives rise to stroma of the cornea, ciliary body and iris, some part of the sclera, primary vitreous, blood vessels, extraocular muscles, optic nerve sheath, fats, ligaments, superior medial walls of the orbit, connective tissue of the upper eyelid. Inferior and lateral walls of the orbit with connective tissue of lower lid are derived from visceral mesoderm of maxillary process below the eye. This diagram is showing the neural plate and the neural groove and the somites. So eye develops at 22nd day of gestational age when the embryo's length is around 2 millimeters and it has 8 somites. At that time, the eye development starts generally. At the 19 days of gestational age, you can see here the neural plate. The neural plate is nothing but on the dorsal surface of the ectoderm. It shows a thickening in the mid portion of the surface ectoderm dorsally. That is nothing but the neural plate, at the center of which there is a groove because of lateral bulging or lateral folding of neural plate. These are called as neural folds. So at the center you can see there is a neural groove and neural folds which are fusing at the center in diagram A. This fusion starts at the center and it progresses at the same time cranially, that is rostrally and caudally. The diagram B is showing the complete fusion of the neural folds. It gives rise to the neural tube formation. So here you can see the cross section through the dorsal surface of an embryo, and it also shows the formation of neural tube along with the neural crest cells. So you can see this E is nothing but the surface ectoderm. NCC means neural crest cells, and these are derived from the surface ectoderm. The surface ectodermal cells, they thicken, they differentiate into neural crest cells at the crest of the neural tube. So how the eye develops? Eye develops from the anterior portion of the neural plate. So this neural plate, it differentiates into various parts later on. So it gives rise to prosencephalon, mesencephalons and the rhombencephalon. So prosencephalon part, it gives rise to telencephalon, diencephalon. Mesencephalon part gives rise to the midbrain. Rhombencephalon is divided again into metencephalon and myelencephalon. I like this diagram for the very good understanding of various parts of the neural plate, how they differentiate into the prosencephalon, mesencephalon, hindbrain, and the structures derived from them. So prosencephalon, it develops into forebrain, midbrain. Mesencephalon, it develops into midbrain, and rhombencephalon, it develops into hindbrain. So prosencephalon, it shows two parts, telencephalon and diencephalon. Midbrain, mesencephalon, it gives rise to midbrain and cerebral aqueduct. The rhombencephalon, it has got two parts, metencephalon and myelencephalon. The telencephalon part of the prosencephalon, it gives rise to cerebral hemispheres and lateral ventricles. The diencephalon part of the prosencephalon, it gives rise to thalamus, hypothalamus, and third ventricle. The mesencephalon, it gives rise to midbrain and cerebral aqueduct. The hindbrain or rhombencephalon, as it divides into two parts, metencephalon and myelencephalon. The metencephalon part of the rhombencephalon, it becomes pons and cerebellum and also forms the upper part of the fourth ventricle. The myelencephalon part of the rhombencephalon, it gives rise to lower part of the fourth ventricle and medulla. So the eye develops from the anterior-most portion of the neural plate, which is prosencephalon. On the lateral sides, anteriorly, the prosencephalon or the forebrain wall, it shows optic grooves on both the sides. These are the optic grooves. They enlarge and they grow laterally forwards to form optic vesicles. These are the optic vesicles. And the proximal portion of the optic vesicle, it narrows down to form the optic stalk on both the sides. This diagram is showing the closure of the anterior neuropore, anteriorly here where the arrow is shown here. These two things, these are optic vesicles. This picture is again about the development of the optic grooves, optic vesicles, optic stalk, and optic cup. This is the diagrammatic representation of the formation of the lens placard, lens pit, and lens vesicle. When optic vesicle, it comes in contact with the surface ectoderm, the area there, it gets thickened to form the lens placard. The lens placard, it starts going inside the optic vesicle and forms the optic cup. The optic cup, it undergoes differential growth in order to engulf the developing lens vesicle. When the lens placard, it starts going inside the optic cup, it gives rise to lens pit when the embryo is 5.5 millimeter length. An optic cup, it undergoes differential growth. It means variable growth. The superior and the lateral parts, they grow more than the inferior part. It gives rise to the formation of choroidal fissure. This picture is again about the formation of lens placard, lens pit, and the lens vesicle. So the lens placard appears at the gestational age of 4.5 weeks. Lens pit appears at 5 weeks and lens vesicle it forms at 5.5 weeks. This picture, it shows the development of the lens. The development of the lens will be covered in other video. So this is the choroidal fissure. It is because of the differential growth of optic cup. This choroidal fissure, it extends from the anterior portion of the optic cup to the distalmost portion of the optic stalk. This diagram is showing the choroidal fissure and the choroidal artery. Normally, the choroidal fissure or embryonic fissure, it gets closed by 6 weeks of gestational age. The process of fusion of choroidal fissure or embryonic fissure, it involves the breakdown of pre-existing junctional complexes and development of new one at the site of fusion. This is the section through the fusion site that is showing the establishment of new junctional complexes between the adjacent retinal pigment epithelial cells, the zonula occludentis and lateral junctions. The significance of choroidal fissure is that failure of fusion of choroidal fissure, which normally occurs at 6 weeks of gestational age, it leads to formation of iris coloboma, ciliary body coloboma, coloboma of the optic disc, retina, and choroid. The coloboma of retina and choroid, they coexist usually because normal development of the retina is required for the normal development of the choroid. That's why they usually coexist. This diagram is showing the iris coloboma. The middle diagram is showing the coloboma of optic disc and retina and choroid. And this one is chorioretinal coloboma. My references for these videos are Wolff's Anatomy of the Eye and the Orbit and Khurana, Anatomy and Physiology of the Eye. This is the last slide on which I have recommended two videos. First video is for the development of the eye and the second video is to understand the basic general embryology. Thank you for your patience.